Okay, so we are discussing some of the molecular characteristics of cancer, and we're going through the some landmarks of cancer. So we are going to continue talking about that, and we're going to start talking about how they are able to replicate forever. Okay, so again, to summarize, we talk about these opposite pathways that are important to growth factor, keep that cells are self-sustained, evading apoptosis. Later we said, okay, now that the tumor grow, and we need food, so you need to have the ability to produce new blood vessels. Now we need to talk about this limitless potential for replication. Yeah, so you not only need to have factors that allow them to survive, but it also need to have the correct changes in the genome that it allow it to do it for a long time. So let's talk about how cells can turn almost, these cancer cells can, if you modify them in the right way, they can turn almost like in stem cells because they have this ability to keep replicating and replicating and replicating. So I'm not going to talk about this because you have heard about this in genetics, and we will cover it in more detail. And I think we cover a little bit in developmental biology, but normal cells cannot divide forever. Yeah, and there is something called the Hay-Flag number. So generally, cells have a specific number of cell divisions, and after that, to avoid, or in multicellular organisms, this happen. So there is something called Hay-Flag number that after a certain number of divisions, they will end up dying. Yeah. And one problem that cells have is the end of the chromosome, and so if you remember, the end of the chromosomes are called telomeres, and telomeres they don't contain genes. Yeah, there's repetitive sequence that are generally lost, but every cell division you can start losing a piece of the chromosome. Yeah, so we I will add a video in case that you wanted to watch it. If not, you can skip it if you remember that. But the idea is that you will start losing some of the components of the telomeres. So the telomeres will start getting smaller and smaller, and there will be a point that you will start losing part of the genes. Okay, so this is why there is replication, normal cells, but they avoid to having many numbers of replication. There is a limit number of replication per cell. Okay, so remember, just telomeres are the natural end of the chromosomes. They have a sequence of DNA that repeat over and over. They exist in different organisms like protozoan, plants, humans, and other ones. And in humans, generally the repetitive section is CCC-TAA, or the complementary, uh, and is repeated between 200 to 1,500 times. Now, if you want to watch this video, here they describe what is the problem that maybe you have heard about this multiple times, so you can skip this part.
The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomere. The ends of chromosomes contain regions referred to as telomeres. These telomeric regions consist of telomeric repeat sequences. The exact sequence of the telomeric repeat can vary from species to species. The repeat sequence shown here is found in the telomeres of human chromosomes. Since DNA replication can only proceed in the 5' to 3' direction and requires a primer, it is not possible for a DNA polymerase to replicate the 3' ends of linear chromosomes. If this problem were not corrected, chromosomes would shorten with each cycle of DNA replication. The addition of telomeric repeat sequences by an enzyme complex referred to as telomerase assures that this shortening does not occur. Telomerase is a unique and interesting enzyme because in addition to having DNA polymerase activity, it also contains an RNA sequence that provides a template for the synthesis of telomeric repeat DNA. Part of the RNA sequence hybridizes with the single-stranded overhang on the DNA strand, leaving a single-stranded overhanging RNA sequence. The repeat unit shown here is from Tetrahymena, which is a model organism used in telomere and telomerase research. The DNA polymerase function of telomerase then synthesizes the DNA strand complementary to the RNA found in telomerase and subsequently translocates to the end of the newly synthesized strand, and the process repeats itself. Many cycles of repeats can occur. Once telomerase has completed its function, DNA primase synthesizes an RNA primer near the 3' end.
Okay, so this is a key topic. Why? Because stem cells are able to solve this problem, yeah, and they have this telomerase active all the time. However, the problem of having this telomerase active all the time is that the cells are never going to die, and this is what happened. This is one of the problems with cancer cells. They can activate this telomerase, so they are all, well, they're going to have now much larger telomeres, and if they have very large telomeres, they will find a way again to replicate forever. So now, to summarize all the things that we have talked about this problem with the telomeres and the end of the replication problem, I want that you remember the following things. First, before cell divides, it replicates its DNA. I think this is clear for everybody. Second thing, the DNA replication is not quite complete. Sometimes a little bit missing is at the end, so at the end of those telomeres, yeah. So therefore, the telomeres will start getting shorter or shorter each replication. The third thing that I want that you remember is that some replication detect when telomeres, some proteins detect when telomeres get too short. And what is going to happen is that when the telomeres get too short, there is a signal, a protein, that say to the cell stop dividing. And however, the problem that we have with cancer cells is that they make more protein, telomerase, which adds more telomere sequence at the end of the DNA. Yeah, so they are just all humans in our DNA. We have that enzyme that is expressed in stem cells, but they are able to now. It's generally only upregulated in stem cells and reproductive cells. Yeah. However, in cancer cells, they are able to turn on that gene and now have the enzyme expressed in the tumors. So this is why the telomeres, the cancer cells, are able to continue replicating without any limit. Okay. Okay. So far we have talked about first thing that cancer cells can do is they create their own growth factor. Yeah. So they are self-sufficient and say to themselves, we need to keep growing. Second thing that they can do is to avoid apoptosis, to abate the signal that say there are pro-apoptotic factors that tell them please stop reproducing and start dying because you have a lot of mistakes. Third thing that they do is they said they keep growing, so they need more food, so they make their own blood vessels to maintain their goal and keep going through, survive, have the food available to keep growing and have a lot of mitosis. Fourth thing that they do is instead of they start dividing and there is a lot of mitosis and there is a limit for division. You know that telomeres start going to get smaller and smaller. Now, what they do is they activate an enzyme that allows them to produce extra telomeres, so they are going to be able to keep replicating and replicating. Okay, so now the fifth thing that we're going to talk today is this insensitivity to anti-growth signals. Yeah, so the fifth thing is that there is growth signals that allow them to stop. Yeah, so like this, please stop growing, and they are going to not be sensitive to those signals. So let me talk about that. Okay, so we're going to talk about this insensitivity of anti-growth signals. So we have here last time that there are signals in our cells, and one of those signals are growth factors that are proteins that say to set signal that say go attach a ligand is going to attach to a receptor that is going to have a cascade of events that is going to end up into change in gene expression. And in this case, the change in gene expression will allow the cells to continue growing and replicating. Yeah. Sometimes you have signals that block these growth factors, and one example of that is this retinoblastoma or RB protein that you have here before. And I'm going to show you a schematic of what happened. And more than telling you the detail is that I want that you remember that we have first growth factors that it will attach to a receptor, and look what happens when it goes inside. It'll activate the RAS pathway. That is not one protein, it's a series of proteins, the MAP kinases, and is going to affect, and I know you studied this in diversity of life, the CDK cycling. If you remember that there is a oscillation of concentration of factors that is going to determine if when mitosis occur, and this is regulated by by RB, by this protein. And if RB is attached to this other protein, there is no transcription. And if there is no transcription, you don't have the right proteins that are needed to go to the S phase, to the synthesis phase. So mitosis cannot take place, yeah, because there is no replication of the DNA. However, in cases when you have this happening, you have a phosphorylation. You see the piece here, the phosphate groups here attaching to retinoblastoma. Now is not able to attach to the E to F, yeah, to this transcription factor. And what is going to happen? Gene transcription, gene translation take place, and now you have all the enzymes and other proteins required to go into the S phase. So mitosis take place. Okay, so what I wanted you to remember from all those things is that you can have proteins that affect other proteins, and those proteins, well, so you have one growth factor that affect the RAS pathway that affect other proteins, that is the CDK, yeah, that it will tell the cells divide or not divide. Yeah, so they are regulators that are important. So there are signals tells the cells to regulate or not. And how this was found? And let me show you a slide of how it was found, this RB. So this mechanism was very important, or is very important, and it was found in one type of childhood cancer. Here you can see that in the iris this translucent structure, that it was found that it was a cancer, a tumor. And later on, we realized that science found that it was change in a mutation in a protein. Just for you to know, between 95 to 98% of these tumors, yeah, of the child that suffer from this cancer in the developed world. Now we have a treatment, so most of them survive if there is the correct treatment, and around 55% of these cases are due to non-genetic form. The other ones are due to inherited form. Yeah, so again, this was the origin, and then they found that it was due to a mutation in this protein that is called the retinoblastoma that control the cell regulation. Now, in the following slide, I'm going to show you how this RB retinoblastoma protein affect the cell cycle through a video, and that summarizes a lot of the information that I have given you.
The cell cycle is controlled by an interaction between cyclins and cyclin-dependent kinases, CDKs. During G1, the cell gradually accumulates cyclin, which binds to CDK to form a complex. When the level of the cyclin-CDK complex exceeds the threshold, the G1 phase ends and DNA replication begins. Cell division is normally turned off in healthy cells by proteins that prevent cyclins from binding to cyclin-dependent protein kinases, CDKs. These proteins are called tumor suppressor proteins. Among the most widely studied tumor suppressor proteins are the retinoblastoma, RB, and P53. The unphosphorylated RB protein binds to transcription factor E2F, a protein which activates genes in the nucleus, preventing this factor from initiating mitosis. The G1 checkpoint is passed when CDK interacts with cyclins to phosphorylate RB, releasing E2F. The protein P16 reinforces the tumor-suppressing role of the RB protein, preventing its phosphorylation by binding to the appropriate CDK cyclin complex and inhibiting its kinase activity. The tumor suppressor protein P53 works at the G1 checkpoint. It senses the integrity of the DNA and activates the DNA repair system if the DNA is damaged. P53 stops the cell cycle at the G1 checkpoint to allow time for the DNA repair by inducing the transcription of P21, which binds to cyclins and CDK and prevents them from interacting. Without the CDK cyclin complex, the cell cycle cannot proceed to the S phase and DNA replication cannot occur.
Okay, so to summarize what you watch in the video, and to continue with the idea, you have these proteins that prevent excess of cell proliferation, and how they do it, because there is this transcription factor that I will create a video, I will record a video for you in case that you don't know what is a transcription factor, but the main idea is that the DNA polymerase need a way to detect the RNA polymerase. It needs to attach here, but it doesn't know exactly where it has to attach, and you have other proteins that allow them to tell, please stop here and start doing your work here, start the transcription, yeah? So those are transcription factors. This is a very general idea of what is a transcription factor. So in this case, you have this protein, this transcription factor is going to attach and tell you need to replicate, stop here and start translator and start transcription here, yeah? So the RNA polymerase is able to start transcription, but it doesn't know exactly where to start, and this transcription factor tells them where to start. So this transcription factor tell the RNA polymerase to start the synthesis, yeah, in proteins that are involved in the DNA synthesis and the cellular replication, as it was shown in the video. So now the S synthesis is going to start. Okay, so now if you have this RB protein attaching to the transcription factor RB, this is a common thing that can happen, that your protein is going to block the a transcription factor. So what is going to happen is that when the RNA polymerase is going to come here and try to attach here, yeah, it doesn't have way to attach. Yeah, there is another protein that is blocking the attachment. So what is going to happen is that this RB, the retinal stemma protein, is going to bind to the E2F, that is going to stop working. It's not going to allow the RNA polymerase to synthesize the protein. So it's not going to, the S phase is not going to take place because there will be no transcription. So conclusion, as I said before, RB prevent the excess of cell proliferation. Key idea, we dissect each one of these steps. So we know that RB, that there will be growth factors that help the cells start dividing. So you go to the S phase. Yeah. However, RB said, no, 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 I don't want that cell division to start. Yeah. So to remove the regulation of RB, the cell need to dephosphorylate something. So the ATP turn into ADP, and look where the phosphate is going to go. It's going to phosphorylate RB, and now is when this can happen. Yeah, so it should be properly regulated. This is the way that it's regulated. When there is a mutation in the RB, yeah, now you are not going to have this attachment all the time. So what is going to happen is that you are going to keep proliferating and proliferating. So you are going to be insensitive to this anti-growth signal. Okay, now I want that you answer the following. Okay, now I want that you think about this. Ways by which cancer cells become insensitive to RB. So I already told you that all the mechanisms, and now I want that you think, okay, but what actually happened in reality? Yeah. So we know that now we know what is the mechanism. Now what happened to a patient that is insensitive to RB, and they could be several things that are affecting. Yeah. So please think about the four, and just to remind you, the human papillomavirus, yeah, inactivate RB. Yeah. So the cancers can become insensitive to anti-growth signals. And what I mean is that there are many ways in which the RB can become insensitive. A cell can become insensitive. So think about four. You can stop the video, and I'm going to give you an answer in a few seconds. Okay, so I'm sure that you wrote this one. The first one is that there could be a mutation in RB. So now the RB cannot attach anymore to E2F. Another thing that it could happen is that there is a mutation, there is a decrease in the expression of RB. So now you don't have enough RB to attach in the correct places to regulate E2F. Another thing that could happen is the opposite, that there is a lot of the transcription factor of E2F. So now you have so many that the proteins, the RB is not able to regulate it. So you break this balance. So you can break it by decreasing RB, or you can increase it by having a lot of the transcription factor E2F. And another way to also break this balance, so here it will be also reduction. And another way to increase it, it will be by producing a lot of growth factor. So if you have a lot of growth factor, you will not have enough RB to start blocking all of them. Okay, so it's not only that you have a mutation in RB, you can be disrupting different parts of the pathway. So again, we are not only thinking about in terms of a single mutation in a protein, but ways in which a pathway can be affected. And here with the RB example, I tried to show you that. Okay, so we're almost done. Let me talk about the last section. Okay, so to finish, we already talk about all of these guys, all these landmarks for cancer, and we talk later. I show you examples of the insensitive to anti-growth signals, and now we're going to talk about another characteristic of cancer that it connects to the beginning of the class. That is the idea of tissue invasion and metastasis that require the remodeling of the extracellular matrix. So let's finish this section. So what we have been talking is that I show you in the beginning all these different stages of cancer. Yeah, and so the idea is that the tumor masses after they grow, so all they grow, they need to go and colonize a place where they have more nutrients and space. Yeah, so they already colonize all those things. So they said we need to go on and grow in other regions. So when that happens, they can move to a different region. So they go and become from a benign, they go and become premalignant or malignant. And to do that, they have to break this basal membrane. Yeah, so the basal membrane here, it needs to be broken, as is shown here. And to do that, we have a group of enzymes that we talk in the beginning of the class. So we have the extracellular membrane. Yeah, that is, as I said, it gives support to animal cells. And here you have the epithelial, the basement membrane, and here you have the extracellular membrane, and here you have the endothelium. So to go from here to here, you have to break these two things, yeah, that are the basement membrane and the extracellular membrane. And the cancer cells make a lot of a protein that is called the matrix metalloproteinases, that just easier to say MMPs, yeah, just how everybody talk about them. So so in a conference you will always hear these MMPs, and everybody knows. So now you know, and you don't have to try to pronounce this very long thing. So and they secrete then in the environment, and so the MMPs are proteins that degrade the extracellular membrane, so the cancer can invade and spread. So here you have this uh, 3D extracellular membrane that we will talk about how producing this 3D extracellular membrane was so important to study cancer and to study the normal function of the cells. So later we will talk about that, and you will hear a little bit of the next lecture talking about that. Okay, to finish this section, I hope that now is clear all these landmarks of cancer. Now you understand it. In the beginning I gave them a very general view. Now you understand it in more detail. I'm sure that you know what now is a tumor suppressor. I'm sure that you know much better what is an oncogene. And if you have any doubt, the videos and the slides are there, and we will keep using that type of those terminology for the following lecture. So now I'm sure that for you is clear that there could be many proteins that could be oncogenes or tumor suppressors that are altered during cancer. Yeah, and now I'm sure that you know that some of these proteins, when they have mutations, they can have be more or less active. Yeah, they can be overexpressed, or they can be a loss of function. But not only you not only have a mutation in the for cancer to appear in that specific protein, but it could be a change in the pathway. Yeah, so you have now changes in the expression, so you can have more or less of that protein, that it could make the cells look relatively normal, but they could be already a problem in those cells. So it's not only a change in the specific protein, but it could be a change in a part of the pathway, that it changed the balance uh, between anti-growth signal or the growth signal, pro-apoptotic or anti-apoptotic. Yeah. So now we know that cancer cells have mutation in their DNA, that some of them, they will be mutated genes in these tumor suppressor or oncogenes, but they could be also mutations in places that cause differential gene expression. Yeah, that cause in the regulatory aspects of the gene expression, so in transcription factor, yeah, that that are very important. So again, I finished this topic of the video lecture, reminding you that now we don't think about chromosomal rearrangements only. We not only think about proto-oncogenes and oncogenes. Yeah, all those are correct. Now we think in terms of pathways, and we not only think about mutation in this specific pathway, but how they are changed the regulation and how they break this balance. And just to finish, remember that you talk in genetics or in our courses in developmental biology, we talk about how genes are regulated, that we have structure like enhancer, promoters, and proteins. If you didn't study this yet, later we can cover it if you have any doubt. But the key thing is that, as I said before, there are many ways in which you can regulate the expression or of the lack of expression of some of those genes. Yeah. So now we know that it's not only that you have a mutation specific here, but there are ways that you can start changing the expression of that gene that it can cause in the long term a change. Okay, so here with this one we finish this section. Now you know we talk about what is cancer, the statistics, and the molecular characteristics of cancer. And if you have any question, just let me know and have a good day. Bye.